Wacht, nee. Rechts, stop. Iets naar rechts. Ja, rechts is tegen. Dicht nog links. Stop. En nu nog weer deze twee. Ja, hoe?
Hereby, I open this academic ceremony in which Reijnders will defend the academic thesis From Pieces to Picture, Systems Biology, Multi-Omics and Machine Learning in Complex Brain Disorders. May I invite you to present a summary of your study and the conclusions of your thesis.
Thank you. Dear co-actor, dear members of the Corona, colleagues, family and friends, I'm excited to stand in front of you today, honestly. To share what I've been working on the last couple of years, and also to share some enthusiasm I feel towards my work. In the following 15 minutes, I will give you an overview of my PhD, of the main findings of my PhD thesis called From Pieces to Picture. To explain what I've been working on, let me start with a metaphor. Imagine that you're handed a box filled with thousands of puzzle pieces, but there's no image to guide you, and after a while, you're not even sure if they belong to the same puzzle. This is what it feels to study complex brain disorders. Each piece represents a different part, like a gene, a biomolecule, or an environmental factor, like stress or diet. Alone, these pieces don't tell us much, but if we can figure out how they fit together, then we can start to see the bigger picture. And my research is all about connecting these pieces, finding the patterns and the relationships that help us understand the whole system. Traditionally, biology has focused on individual factors to understand how they work, like a single gene or a molecule. And while this is very useful, they can sometimes miss the bigger picture. And this is where system biology zooms out and looks at how all factors work together, which is essential in complex brain disorders. These complex brain disorders are not caused by a single factor, but are resulted from many small genetic factors interacting with the environment, like stress and diet. This means that individuals might experience similar events, but their risk on developing this complex disorder varies, making these conditions especially difficult to understand. And in my work, I focus on two complex brain disorders. These are post-traumatic stress disorder and Parkinson's disease, showing how system biology can be used to better understand these conditions. While system biology is the way how we want to study the bigger picture, we also need the right tools and measurements to do so. And this is where omics comes in. Biology itself can be separated in different layers, called omics. Each layer provides specific information, but together they give a clear understanding of the underlying biology. I focus on 3 of these layers. These are called genomics, epigenomics, and transcriptomics, which I will address in the upcoming slides. Genomics is the study of an organism's complete set of DNA, also called the genome. It focuses on identifying the genes, understanding their roles, but also exploring how they interact. And to draw an analogy, you can see the genome as a book you want to read. Important in genomics is studying the DNA differences, as these can influence disease risk, but also response to treatment. By studying the DNA differences, it helps us understand diseases better and develop treatments that fit each person's unique genetics. Epigenetics looks at how genes can be switched on or off without changing the DNA itself. To continue this analogy, it's like opening the book and selecting a page you want to read. A specific epigenetic mechanism I studied is called DNA methylation. This is because environmental factors like stress and diet can have an effect on DNA methylation on a molecular level. It shows how the environment can have a direct effect on our genes and impact our health. Transcriptomics looks at which genes are active at a given time. It shows which biomolecules were produced and that result in protein production. Analyzing gene activity helps us understand how cells, for example, can respond to different conditions like stress or disease. It reveals potential biomarkers, but also treatment targets. And to bring it back to the analogy, it's like reading the text on the page. By connecting all these omic layers, it reveals how these regulatory mechanisms work together and are dependent on each other. And we call this multi-omics. Together, DNA methylation plays a key role in turning genes on or off, regulating their gene activity, guided by the genome's instructions. However, DNA differences can also influence how efficiently DNA methylation occurs, while also directly impacting the gene activity by changing the genome's instructions. This highlights the interaction, the complex interaction between the environment and the DNA differences, which in turn affect the gene activity. Capturing the data, the regulatory mechanisms, and exceptions isn't simple. It requires analyzing massive amounts of data, which is both challenging and time-intensive. The scale and complexity of the data make it one of the most demanding and powerful tools to understand biology. And this is where machine learning comes in. Machine learning is a type of technology that helps find patterns and connections in complex data, but much faster and more efficiently than a human ever could. Well, now I've explored the concepts central to my work. Let me also show you how I apply these. The first part of my thesis, I focus on post-traumatic stress disorder, which is a condition that is triggered by experiencing or witnessing a traumatic event. It leaves individuals suffering with intense emotional and physical responses. In chapter 2 of my thesis, I worked on a blood-based analysis of why some soldiers develop PTSD. More specifically, my research focused on understanding why some of the soldiers show PTSD symptoms, which we call susceptibility, while others do not, even after facing similar levels of trauma. To study this, I analyzed multi-omics data from blood samples, and this work had three main objectives. The first aim was to measure the gene activity specific to individuals showing susceptibility to PTSD, compared to those who are resilient to PTSD. And in doing so, I identified 21 genes linked to PTSD susceptibility. And interestingly, many of these genes were involved in immune responses, confirming that the immune system and stress interact in susceptibility to PTSD. The second aim of my research was to explore potential genetic and epigenetic mechanisms regulating the genes of interest. I identified 64 DNA methylation changes that play a role in regulating gene activity. Additionally, I also found 2 genetic differences that show gene regulation of these genes. For the final aim, I investigated if PTSD symptoms can either be affected directly by the observed regulatory mechanism, either the DNA methylation or the DNA differences. And among these 21 identified genes, only one gene was highlighted. This gene is called TBC1D16. Its gene activity was influenced by a specific genetic difference and in turn contributed to the symptoms indicative of susceptibility to PTSD. The study in chapter 2 highlighted the importance of blood-based biomarkers and also the genetic factors enhancing our understanding in PTSD. These insights, they contribute to the development of predictive tools and the treatment strategies aimed at reducing the impact of trauma. In chapter 3, I deepened my investigation into DNA methylation. The aim of this study was to determine whether DNA methylation serves as a mechanism connecting trauma exposure to PTSD-like symptoms. These samples were obtained from the inner lining of the cheek, making it very accessible but also very non-invasive. Some of these individuals in my study showed PTSD-like symptoms after being exposed to trauma. And I hypothesize that this can be partially explained by DNA methylation. That means that trauma exposure affects DNA methylation and in turn causes PTSD-like symptoms. And in my work, indeed, I found that trauma exposure led to specific changes in DNA methylation, linked to, and some of the changes were partially linked to the increase of PTSD symptoms. One of the most interesting findings was a DNA methylation change near a gene called PCSK2, and this gene is linked to the processing of stress hormones. This chapter shows how stress can leave an environmental fingerprint that affects mental health. By identifying this fingerprint, we improve our understanding of PTSD, making it possible to identify at-risk individuals earlier and explore targeted treatments. And additionally, also develop simple, non-invasive diagnostic tools. The second part of my thesis addressed Parkinson's disease. While Parkinson's disease is widely recognized for its motor control symptoms, caused by degeneration of specific brain cells in the brain, my research concentrates on a less visible but equally significant aspect, and this is the functioning of memory and thinking, as it often gets worse as the disease progresses. In Chapter 6, I employ machine learning methods and techniques to predict changes in memory and thinking performance over time. These memory and thinking problems are a major concern for people with Parkinson's disease, as many develop severe memory issues or even dementia within 10 years of diagnosis. However, predicting who is at risk has been a persistent challenge. In my research, I use advanced machine learning techniques to analyze a group of new PD patients to predict who will develop memory problems and thinking problems over an 8-year period. And to do so, I use a range of baseline data that included genetic and epigenetic factors, but also biological and clinical tests. Using 4 different machine learning algorithms, I developed models to accurately identify who's at risk. These machine learning models are often referred to as black boxes because their decision-making processes are not easily interpretable. But to address this, I applied techniques to make these models more transparent and understandable, to see which of the pieces are really important. My findings revealed combining clinical and biological data produced the most accurate predictions, although clinical data alone performed also very well. Because of the efforts taken to make the models transparent and understandable, it was possible for me to identify which pieces provides the most important information to make an accurate prediction. These results show that combining different types of data helps in the identification of people with memory and thinking problems, leading to an earlier diagnosis and better personalized care for those with Parkinson's disease. In conclusion, my PhD thesis demonstrates how integrating multi-omics data can explore the complex biological mechanisms underlying PTSD and Parkinson's disease. So, why does this research matter? First, it deepens our understanding of the root causes of the brain disorders. By identifying the pieces, the genes, and the interactions involved, we begin to discover the biological mechanisms at play in conditions like PTSD and Parkinson's disease. Second, it promotes personalized medicine, a future where treatments are tailored to a unique individual's need rather than a one-size-fits-all approach. This research is a significant step towards making that vision a reality. And third, this knowledge holds the potential for earlier diagnosis. By identifying individuals at risk before the symptoms develop, we can intervene sooner and also potentially preventing or avoiding the effects of these conditions. Which brings me back to the pieces. In my work, I've uncovered some small but important pieces in understanding PTSD and Parkinson's disease. It offers valuable insight, but there's still a lot more to explore. The ultimate goal is to turn these scientific insights into practical solutions, whether this is through new diagnostic tools, targeted therapies, or a deeper understanding of the human brain. even if you could assemble the whole puzzle right now, it would only show an image of something much larger and much more complicated, something that we don't fully understand yet. It's just the beginning of a much bigger journey. And with this, I would like to thank you all for your attention, and we'll give the word back to the prorector.
Thank you for this very clear presentation of this very complex topic. As a hematologist, I was able to understand at least your presentation, and I hope the audience also enjoyed it. But now we will start the opposition, and the opposition will be opened by Professor van der Hoven, Professor in Neuroepigenetics at this university.
Thank you very much. First of all, my compliments for the very nice thesis and also your presentation. Also my compliments to your supervision committee. Ook complimenten voor de Limburgse samenvatting. Ik had hier de vraag in het Limburgs gesteld, maar dat mag niet. En dat samen met het feit dat mijn vrouw dezelfde achternaam heet als die, als u, sorry. Het zou zeker tot problemen leiden. So I switch back to English. In terms of asking my questions, let's start on chapter 2, if I may, where you discuss the strengths and limitations of your approach, particularly when it deals with power issues. And you, as part of your answer, how you try to compensate for the lack of power, you talk about using multiple layers, which you also explained in your presentation. And I would like to engage a discussion with you because I would argue that it doesn't necessarily increase power. Let me give an example. You take a sample from the population. By coincidence, you have people with a certain methylation profile. Well, you actually show that methylation correlates with expression to a considerable degree. So coincidentally, well, not coincidentally actually, you also have people with a certain expression profile. So how can you convince me that adding those layers of information would, in this case, compensate for the lack of power? How do you see this?
Esteemed opponent, thank you for your comments and your congratulations. I really appreciate it. Also for the Limburgse samenvatting, also. I agree. Power is a severe issue, especially in a study that is effectively 20 versus 20. How do you raise this? How can you conceptually raise the power to a degree where your differential expression or any of these other layers show significance after correcting for multiple testing? One way is indeed like increasing the sample size, but the other point is also like, what if we had biological information or informed layers, as stated by the cisbio concept? What if you were to compound these layers, this information? As you also noted, the level of expression and DNA methylation, they need to correlate in a certain way. So to what degree, what is the coincidence and what is the true signal out there? And while I'm being lenient on some of these layers, I also think that by looking as broad as possible in some steps, we can indeed increase the sensitivity by looking at other compounding information, leading to maybe still in the end a lenient perspective, but will give us more stronger leads in the direction that we want to go.
Okay, thanks. Let me switch gears to chapter 3 then actually, where you did a mediation analysis and you actually nicely also show that you look at methylation changes over time and then link that to changes in PTSD-like symptoms. And my question is, does mediation in this case guarantee causality, or to which extent do you think it represents or reflects causality? Because some of the symptoms, some of the methylation changes may also occur in response to the development of symptoms, and they might not necessarily have to do with the pathogenesis of the disease. How do you see that?
I think there's a difference to be made between susceptibility and diagnostics, with having accurate predictors. In this case, the mediation analysis the method itself is built in such a way that it can prove a causal path. But indeed, to what degree is it influenced? To tackle this, I mean, in this study, again, there was quite a limited set of samples that were evaluated. The types of trauma were quite diverse, or stress exposure actually. The types of phenotype or the outcome that was used is also to some degree, I would also say, quite lenient. So there's a lot of variance there that might account or not account for the changes observed. However, this mediation analysis is quite, I would say, strict in terms of like passing the criteria, especially the last part where it applies the bootstrapping period, bootstrapping process. In this point, it really this will leverage the information that we can sort of state like, okay, is this truly mediation or not? But I'd like to note indeed that while we speak of mediation here, all of these CPGs are indeed partially mediated.
Yeah. And of course, if you have lots of power, you would ideally also look at methylation signatures before exposure and see whether they can predict. Huh? Okay, thanks. Then maybe a final question, switching to, well, actually statement number 7, if one of your paranames could read out statement number 7.
The interplay of the genetics, epigenetics, and environment suggest we conceptualize stress disorder like PTSD, neurodegenerative diseases, and dynamic phototypes.
Okay, thank you very much. I think I agree, but at the same time, I also see huge differences between the two classes of disorders, well, PTSD and the neurodegenerative disorders that you describe. Because, for example, Alzheimer's disease, yeah, there seems to be a point of no return. You cannot sort of recover from that unless we find a treatment that does the job. Well, with PTSD, it seems more dynamic. So how do you see that, and why did you put them on one pile, sort of, in terms of the statement? Why did you regard them as similar for the statement?
I'm sorry, could you clarify a little bit?
So they are both dynamic, but at the same time they are different. PTSD from neurodegenerative disorders like Alzheimer's, for example. Because with Alzheimer's, you have a gradual increase in pathology and all these processes, and then there's a turning point, and sort of after that turning point, unfortunately, with current treatment strategies, you are doomed. With PTSD, it changes over time, but you can actually also sort of recover from that, hopefully. Yeah. So how do you see that? Do you see the same dynamics or not? What's your view on this?
I think in the bigger evaluating everything as a system with dynamics, which we up until now we have not really done so, right? We've only taken snapshots before and after, or like after indeed a certain period where there's a point of no return, as you state. There might be changes in other phenotypes or other outcomes of neurodegenerative diseases where these systems might go slightly out of balance, and then from that point you might enter this point of no return. But I think for now the current methods, unraveling this specifically would require having dedicated studies that also include the temporal aspect very strongly, way more in detail than we have done before. So on that regard, while PTSD is a clear environmental stimulus that causes the PTSD and can be recovered from, I think in terms of dynamics from the bigger picture, I would see similar kinds of behavior, but maybe not in this moment of time where we have the tools to measure to do so. Okay.
No, I fully agree that longitudinal follow-up and sampling, etc., would be of great added value. Thank you very much. I'm happy with the answer. I'll give the word back to the program.
Thank you. The opposition will be continued by Professor Martinez, Professor of Neuroinflammation and Neuropsychiatric Disorders at this university.
I would like to start congratulating the candidate and the promotion team for a very interesting PhD thesis. I think I really enjoy and have many ideas already by starting reading it in which things you could investigate. But today we are here to ask you questions. So I would like to start with questions regarding Chapter 2. So in Chapter 2, you kind of look to the blood, right? So when you look to the blood, of course, what do you find? So which are the majority of the cells in the blood?
They're going to be immune cells.
Immune cells. So, and specifically from the immune cells, which are those cells where there is genetic information present? Because you have immune cells, you have blood cells, for example, and you have lymphocytes. So you don't have genetic information in the blood cells. So you are 90% of your information comes from lymphocytes in the blood, right? Mm-hmm. So in fact, you are looking to a biopsy of the immune system. So do you have examples from biology where the immune system is affected by stress? So what is changing in the blood that can activate the immune system?
Mm-hmm. Alistine Important, thank you for your comments and your question. Indeed, I think the immune system within the context of PTSD is totally dysregulated because of dysregulation of the HPA axis. Cortisol and other hormones, they are enhanced, they are increased over time, or decreased, depending on which. But these were all. This whole process will start a signaling cascade that will infect the immune system on all levels, not only on, for example, the lymphocyte quantity, but also like their intrinsic patterns, maybe even the DNA methylation that has been observed. This will start a whole cascade, effectively.
Yeah, so thanks for that. So I was also looking to the amount of genes that you have found dysregulated, and you will have a question about this later on. And looking to these genes, there is one in particular which is in fact unregulated, that is TBC1D16, which you look into the biological functions of this gene. It seems to be quite broad in terms of affecting probably affecting many cells of the body, so it's present in different cells of the body. So do you have have you found any relationship between this gene and, for example, cortisol?
Well, in this study, I didn't have any direct measures on cortisol. The other chapter that I worked on, indeed, I also looked at this gene, but this was not differentially expressed or whatsoever. So on that case, there was no direct link that can be found, although I can understand if I look at the potential mechanism behind this, that it can be affected as such. But then the question to me also comes a little bit like, is this now correlation or causality on that point?
Yeah, so, and then I would like to continue asking you regarding your last statement. Perhaps the Paranim could read it, so statement number 10, which I really like, to be honest. I'd like to know why you choose it.
Imagination is more important than knowledge. Knowledge is limited. Imagination encircles the world. Albert Einstein.
Okay, so did you choose this statement based in any of your chapters in particular? That's something that you really had to use your imagination, and then you have a very striking finding and you are very proud of? Could you explain this?
I'm definitely very proud of this, but it has actually nothing to do with my thesis, to be honest. I actually got it as a present for my parents at the time, as like a little frame from finishing my master thesis, my master education. However, I strongly agree. Well.
Okay, yeah, I used this statement to use my imagination in your thesis, and then I found that the gene TBC1D16 is very interesting because it's regulating a virus that is some retroviruses that are integrated in the genome. So in fact, the levels of this gene affect whether the retrovirus gets activated. And in fact, when this gene is downregulated, it will probably activate the retrovirus. So just a question to you for the future could be to analyze that in those individuals that have this downregulation. But now moving on to a next question, I would like to know, when you look to all this data, you have sometimes a problem of power, right? So you don't have sometimes enough individuals to really answer all these questions with all these layers that you were asked about. So do you think that perhaps to choose, instead of a broad analysis where you pick up everything is there, if you will be targeting a specific pathway and then you will do a targeted approach, will perhaps lead you to a more detailed knowledge or will precise knowledge. What do you think about that?
Absolutely. No, I fully agree. In terms of targeted analysis, these are the, in my eyes, the most straightforward paths to take, especially after having generated such broad data that highlights some directions of interest. I think looking specifically into pathways, at least where causality is known to some degree, will really give us a nice opportunity to see to what degree can we are the true effects of the genes that we found or are of interest.
Okay, I don't know if I have time left. A small question then regarding this, and also regarding the question about dynamics. Well, when you look to cells and when you look to phenomena, mechanisms, pathways, the cells are dynamic, they are not static. So do you think that your approach is dynamic enough to pick up dynamic changes?
Well, I've learned that indeed these studies are great starting points, but yeah, we definitely need more time points, especially if you look at, for example, at some of the other chapters in chapter 4, where I try to perform one of these trajectory analysis. It's quite limited, but gives us good indications on how to work, on how these dynamics work.
And when you look to the artificial intelligence analysis that you have performed, do you think this has still limitations? What will you do if you will have enough time and resources to continue? Will you continue with them, or you will use other type of analysis to interpret the data? Mm-hmm.
Depends on which direction the study goes. If this is a very broad explorative study, then I would still initially go for the more statistically known, like differential expression or pathway or this kind of analysis. If it is going to be targeted studies where dynamics plays a key role, I would try to incorporate as many time points. But with AI and with machine learning, these things are these methods are really good in terms of predictive power, in terms of finding exactly which variable is now really important. And I would fit them in the studies that question needs to be answered. Also, one of the points that I want to raise, like, okay, machine learning is great, it's very strong, but do you want it to be predictive, or do you want it to be insightful? And I really emphasize that there needs to be some sort of intermediate where we balance both.
Okay, thank you. I am satisfied. I give the word back to the prorector.
Thank you. Your position will be continued by Dr. Daskalakis, Associate Professor of Psychiatry at Harvard University. Thank you for joining from Boston. I'm afraid this. Okay, he's still there.
Good afternoon. Hi, can you hear me?
Yes, we can hear you.
Yeah. Good afternoon from Boston. Rick, congratulations for your thesis. It's a very nice body of work, and also I want to congratulate you of succeeding to combine multiple neuropsychiatric disorders in one thesis that they have commonalities but also significant differences. So I think it was a nice, coherent work. I know that it was hard to manage to do that. I want to start with my first question, and it relates to chapter 5 and chapter 6, that one of the common clinical outcomes that you study is cognitive impairment. And in chapter 5, you are using a variety of GWASs as a source to derive polygenic risk scores, and then you prove that, for example, specifically for cognitive impairment, that it is, based on your statistical analysis, it is useful to use these multi-domain polygenic risk scores. Your model requires weights from all polygenic risk scores that you had in relation to the other outcomes. And then I want to contrast this in relation to chapter 6, where you have this as an outcome, but then you try to see if you use genetic information and or other biological data and or clinical information, what happens. and you see that the AUC is with genetics only quite low, moderate, and then basically clinical data alone is the most critical factor, and then by adding all the biological information, you have a small improvement of performance. So this makes me wonder, what does it tell us about the strength of these different features? And is this the type of analysis that helps us understand biology, or is a plain statistical data science type of approach?
Esteemed opponent, if I understand your question correctly, you're asking me specifically to what the variable diversity contributes, right? Okay. Well, indeed, and as you rightfully state so, in Chapter 5, I look at these multi-trait polygenic scores, where these are built up from multiple different GWASs, dependent on the outcome, dependent on which GWASs were available, which variables were available. The prediction became either, there was in general like either no improvement or a slight improvement, and in this case with a cognitive impairment, because we do not have a dedicated cognitive impairment GWAS, at least at the time when I was running it. It could be approximated by the combination of all the other GWAS, a little bit better than trying to make a proxy using, for example, the Alzheimer's disease GWAS study used. In that process, I discovered indeed that all of the, in general, like all of the variables, they seemed to either not give a strong contribution, and I was genuinely wondering, okay, why is this not the case? Because effectively, you would say like adding more variables, adding more information gives also the machine learning the time and the process to, or at least the potential to learn better, to make more accurate predictions. But if these variables were not correlated or strongly correlated enough with the outcome, these were effectively included as noise. So that's one part of the chapter 5 that I realized. But also in chapter 6, there we had very strong correlative variables all over the study. So when starting out with even 200 or 400 variables, it all boiled down to maybe this 30 that were some of them were highly correlated, showing high multicollinearity as well. But then still adding the extra part of genetics, adding the extra part of the DNA methylation, still managed to make a slight improvement. And I think because these variables specifically were somewhat still correlated and had some novel information towards the general outcome that I wanted to predict. What I learned here in these steps is that while adding everything on machine learning sounds great, you need to be smart about what and how should you tackle the dimensionality problem. If you're just going to go with, okay, we're going to select all the correlated ones, then you might also miss some variables. So you might want to use some other metrics to evaluate this interaction, so like effect.
So do you see opportunities by using a different nonlinear approaches in your method for capturing, let's say, even more benefits of adding those variables, the non-correlated variables into this? Let's say if you would do it, would you change the machine learning approach to maybe boost even more with genetics by incorporating a nonlinear model or something like that?
The machine learning models that I in the end selected, these were quite robust in terms of which models to be selected. And some of them, they were nonlinear. If you think about, for example, the random forest one, or even the support vector machines, although using linear kernels doesn't make it specifically nonlinear. But for taking the example of the random forest, you could also, I didn't try this, but you could also apply to only train random forest models on the genetics part, trying to make the best predictors. Maybe there's some underlying nonlinear interacts within the genetics, or maybe adding some other variables that we see that that we know of that they have some interactive effects. But I try to incorporate a broad set of machine learning models that were able to capture linear-like and also non-linear-like patterns in the data. Although in the end, everything also comes back to the outcome, to the phenotype we want to predict.
Thank you. I want to move to my second question, and it relates to another chapter, the chapter 4, that is focusing on the glucocorticoid. Glucocorticoids in relation to differentiation in a neuronal culture as a model of stress-related disorders. I wanted to bring in your attention that cortisol exerts interactions through the glucocorticoid receptor, most likely in this system. And in your analysis, you studied the interaction between cortisol and cellular differentiation. And maybe mainly you interpreted that the direction of effect is cortisol affecting differentiation. But if you bring in the glucocorticoid receptor in the discussion, I was wondering, how do you interpret your findings? And have you considered potential fluctuations of the receptor happening too? So is could it be partially those interactions explained by some dynamics of the receptor, influenced by differentiation itself? So basically, and I think because in your thesis you have, I congratulate you that you tried also causal approaches to understand this type of topics. I wonder if you want to kind of discuss a little bit about that difficulty in this study.
Can you give a short answer, because we have 3 more opponents to go, and you want to enjoy also their discussion points, yes. Isn't it?
Absolutely. To give a short answer, in the work that we performed, we only had 3 time points. So in terms of dynamics, it was quite limited that we wanted what we could evaluate. If effectively these 3 time points brings it back to like more differential expression like analysis or interaction analysis. But indeed, this could be altered by the dynamics of the differentiation. Indeed, but this is something we didn't totally investigate, and also we didn't have the resolution to look into that specifically at this point.
Okay, thank you, Dr. Daskalakis. We continue. In real life, people are able to give short answers, but during a defense like this, it's always a bit...
Absolutely.
And that's good. That's very good. The opposition will be continued by Dr. Adriaans, Assistant Professor at the Maastricht Center of Systems Biology.
All right. Dear candidate, first and foremost, congratulations on a very nice thesis. Also, congratulations to your supervisory team. I read it with great pleasure. It demonstrates that you have gained a lot of skills and that you also have a systems level way of thinking, but then I would expect nothing less of a systems biology alumnus, of course. So you picked apart, digitally at least, people's brains, and now I want to pick your brain for a little bit. And I want to start with chapter 2. You mention in the method section very briefly that you corrected for cell type composition. And you refer to supplementary materials, which unfortunately I could not find. So maybe you could elaborate on what method you used and why you chose to correct for this in the first place.
Esteemed opponent, thank you for your congratulations and your comments and your question. Yeah, indeed, that is true. One of the little drawbacks and didn't include supplementary, but then it would have been a little bit bigger. I agree. The self-type correction method, we used Serval, actually. One of them that we applied was based on the DNA methylation, to basically use an epigenetic, not clock, epigenetic model to determine the cell-type proportions. And then effectively, I think I also tried to do do something similar with the expression levels. I'm not even really sure if I put all the methods in the supplementary methods, but I tried a lot here to just see. But one thing that I observed was that all of these methods, they were varying quite a lot. I found it a little bit. I found it actually a risk to try to correct for these factors that, if properly calculated, they should give the same proportions, but they didn't. So at this point, I wanted it to be as open as possible. So I just gave the results uncorrected by cell type.
Mm-hmm. And did you observe differences in cell type composition from these algorithms or not?
Yeah.
And in your opinion, was that biology or was that indeed confounding? In this
confounding or data-driven, yeah. I think the most strong predictors that we have are still the epigenetic or the DNA methylation models, as these are clearly DNA methylation is indicative of cell lineage, cell differentiation. So these can easily assess the proportions if you have the measures well. At this point, I also was wondering, honestly, in this point, I was wondering, do these measures come from the same samples as well? Because if you take a different sample and don't do some proper treatment of the samples, then also these might differ from your observed difference expression results, or trying to correct for them.
Yeah, you mean if the RNA-seq data and the DNA methylation data are literally from the same sample with the same composition, you might indeed overcompensate. Yeah. Then you do the differential gene expression analysis. You end up with 21 differentially expressed genes. You're a little bit strict, maybe on the fold change there, but that's not a lot, right? Were you disappointed?
No, it was as expected. Looking at this phenotype, it is so heterogeneous. From the samples that we have, 20 versus 20, it's low. I did some sensitivity analysis, not shown in there, but it shows that it's a little bit, I would say, all over the place. But the effective the sensitivity of this was quite how to say this? Was not the greatest to observe.
Mm-hmm. But you do use that as the basis to integrate the DNA methylation data. You also use data from public repositories to kind of enhance the biology underlying it. And about this, like the gathering that data from online resources, aren't you afraid that you just, establish things that are already known? Since you have only a few results, and then you just dig and dig until you find certain relations, and then look and behold, here's biology. Is that a risk, or do you think that this actually is meaningful, that what you've found?
What I think is that it actually is meaningful. Yeah, that is true. The risk really comes in, I would say, like trying to. Trying to publish, or trying to publish the same kind of results. What I specifically use were then indeed the EQTLs and MQTL information, which inherently should be similar given the similar cohort or population. By doing so, I don't think I would be confounding or adjusting my results in any way. That is not biological. I say it like this.
Okay. Do I still have time for a short question?
No.
I'll see you later.
I'm sure you can continue for hours, but the time is limited. The opposition will be continued by Professor Homberg, Professor of Translational Neuroscience at the Radboud University Medical Center. Thank you for joining from Nijmegen.
Dear candidates, congratulations with your thesis. I've read it with pleasure, and I found it written in a very clear and accessible manner, and that really pleased me because I'm not totally an expert in your field, but I could really understand it. And of course, also the topic of understanding the omics of susceptibility to major brain disorders is very important. And I also like that you targeted the omics at very different levels, the epigenetics, genetics, transcriptomics, and also use biosamples of patients, but also cell lines. So yeah, you used a lot of disciplines. So my compliments, and of course also for your supervision team. I also have a few questions. The first thing that came into my mind when I was reading your thesis is why did you actually focus on PTSD and Parkinson's disease? So of course, these are major disorders, but also very different disorders. So PTSD is a psychiatric disorder. Parkinson's disease is more a neurological disorder. The origin of these disorders are very different. So why did you pick these two?
Esteemed opponent, thank you for your comments and your question. Yes, I agree. I think this is one of the most difficult things that I wanted to work into my thesis, effectively. It is a different way how, if it only was PTSD, for example, then I could have made a thesis from beginning to end and it would have been very clear and such. This, why I included Parkinson's disease or the cognitive impairment part, was a part of practicality and also learning. So I had a great ambition to also perform my machine learning methods, my machine learning ideas. And for this, also needs a very strong phenotype or very clear path, let's do it like this. And in discussing this, I was able to work on a project that also, I passed Parkinson's disease as well because of that, effectively.
Okay, that is also just how it sometimes runs in real life during research. Okay, thank you. Then I wanted to ask a question about chapter 2. Epigenetic modifications are known to be very local. So first I want to ask how do blood cells or epigenetic changes in the blood cells reflect changes in brain mechanisms? I also read that in chapter 3, you actually addressed it by focusing on buccal cells, and I found it very smart. But of course, still there are very regional differences in different brain areas. There can be different epigenetic modifications, and even in different cells, there can be very specific modifications. So what do we learn from the brain by the blood or the buccal cells?
Mm-hmm. Yeah, this is, I think, a very relevant question. For me, looking at the blood cells, at the immune cells, trying to make a translation to the brain is inherently difficult, as you already given the proper reasoning to do also. In my eyes, I think we should have more correlative analysis to figure out, okay, how does indeed one DNA methylation profile from one cell type can be affecting truly some other cell region. But in the studies that I have, I was also limited to its tissue types and to its designs. So on a specific question, on a specific answer on how will this specifically interact, I cannot answer at this given time. But I would only imagine that this would require great efforts in intervention-like studies.
Okay, thank you. Do you think it might help to combine human and animal research? So, for instance, if you have both in animals, of course, we can go a step further, and then you can collect brain tissue. And if you compare the data from the two species, for instance, both species, you collect blood and you measure the epigenetic modification. Of course, you need a very good animal model that I agree about. But might that be an approach to understand or have a better idea of what the link between the blood and the brain is, do you think?
Sorry, I didn't capture the last.
Yeah, but I just wanted to ask if you agree with such an approach, if that would be helpful.
Yeah, absolutely, absolutely. No, I think this translation needs to be done to some degree. It is, I would say, almost highly unethical to do like PTSD, like intervention studies. This is something that needs to be simulated to in an organism that is as closely as possible, closely relatable, that we can have, that we can assess. So indeed, doing this animal studies, we might be able to come a lot closer to the true interactions or the true effects that for example, the DNA methylation could have. Yes, I agree.
Okay, thank you. Then I have a question about chapter 4, which is about the chronic cortisol exposure in human embryonic stem cells. So I was curious if the embryonic changes in gene expression can really predispose to negative effects or trauma in later life. Is anything known about that?
I'm sorry? Could you repeat the question?
So my question is if embryonic changes in gene expression or epigenetic modification in the embryonic phase, can that already predispose to negative effects in later life, like more susceptibility to trauma or more susceptibility to the development of PTSD? Do you know whether there is any such evidence, or it's very hard to investigate probably, but perhaps you know anything about this?
I think there's definitely reason to at least to look into this. There are enough. I'm trying to recall exactly. I cannot recall exactly right now which studies investigate this, but yeah, there would be any predisposition that could affect later life. In my study that we ran, we only looked at the cohort interaction in the cell type differentiation. Here we could see that the effects of cortisol have at least great effects on very critical points in the differentiation itself. So how this will manifest later in later life is something to be investigated, but I think these implications would definitely be biologically relevant.
If I may, can I continue? Last one question.
I'm sorry to interrupt you, because all the opponents are so enthusiastic they can discuss for hours, I'm sure. Okay. They're all experts. The opposition will be continued and finalized by Dr. Driessens, Associate Professor at the Department of Advanced Computing Sciences.
Thank you, Pro-Rector. Dear candidate, congratulations on the dissertation that you put in front of us, also to the supervisory team. Yeah, I have questions on chapter 5 and 6, which will not come as a surprise to you because they'll deal with machine learning. The first one that I have is on your choice of evaluation measure. So you have a number of them, but one of the ones that makes a big deal, right, of this evaluation is the area under curve in the ROC curves. Now, this was invented by machine learners who wanted to show, hey, my new algorithm works for a large number of cases. Look, I can summarize a lot of different tests and then show you this value. However, you were answering quite specific questions. And if you look at these ROC curves, for example, on page 150, you see that sometimes they can leapfrog depending on where in the space we are talking about. And these ROC curves allow you to determine which algorithm, under even which setting, is best for a specific cost of false positive and false negative predictions. And so I wonder, would it make sense to do a deep dive into that? Is that very difficult? Why did you not do it? Can you comment on that?
Esteemed opponent, thank you for your congratulations and also your question. Honestly, I was struggling with this myself as well. I think the evaluation metric is one of the most important things. It will indeed state like this model is good, this model is bad. My results are good or my results are bad. What I investigated at the time is also not really mentioned in here. Is like I put all of these different metrics not next to each other, but then comparatively with, I think at the time it was still random forest. I just wanted to see like, okay, how do they behave and how do they behave in certain conditions? Like I want a model that says no when it is required to say no, rather than just say no because 99% of my population is no. So this is what I went into. And then effectively I found that this matches correlation coefficient is quite interesting because it tries to correct for that imbalance, I would say.
Okay. My second question that I want to ask is about the choice of models that you made. So you said before that your choice for SVMs with a linear kernel stays within a linear model. In chapter 5, you have like carried nearest neighbors, a Bayesian approach, random forests, and then the elastic nets, which is a linear model. And in chapter 6, you replace KNN with SVMs, and you have 2 types of forest and then 2 types of linear models. So I have 2 questions about that. First one is, why did you decide to include linear SVMs as an alternative to elastic nets? What do you think this brings? And second, why not include more complex kernels, like a radial basis function or something like that?
Yes, we went for linear kernels for a very specific reason at the time, because we wanted to make these machine learning models as open as possible, so we can really assess what these variables, which variables are important. And by using nonlinear kernels, the translation to make this assessment of which variables are really important becomes more difficult. So in the end, that's why we went for the support factor machines and not for different kernels as well. But yeah, I agree. The choice of machine learning models really has a great impact and its eventual performance as well.
Okay, and what do you think a linear SVM does differently than an elastic net?
You can give a short answer, but it's not necessary.
I can give a long answer? I can give a long answer?
Yeah.
That a linear kerneled SVM is similar to elastic net. Intrinsically, and trying to recover exactly how the SVM works, but I must admit I never understood how they were raising the dimensions to a higher power compared to ElasticNet, which is just the regularization metric that is added or risen to a power of x.
This is not a short answer.
Oh, sorry.
Like all your opponents, you can also continue for hours and show your knowledge. That's great. Rick Reijnders, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. And I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Southside, 7 miles in my rearview mirror. I know what it felt like. My goals only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the line, I know you know that I can't read time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. And even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. South side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Rick Reijnders, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the committee has decided to grant you the degree of doctor. Professor Rutte is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Okay, then. By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Rick Antoine Reijnders, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. So, and for the laudatio, I would like to invite the formidable co-promoter, Dr. Eisen, to start off.
Okay, thank you. Actually, both of us will address you, Rick. So, yeah, I'm happy to be able to congratulate you as one of the first. So I'll do that. I include your family and friends, your parents, your wife, Sarah, and maybe your brand new family-in-law. I don't know whether they're listening online or not. Okay. But in any case, if they do, congratulations to them as well. I don't know where to look. There's cameras all over the place. Okay, for those who don't speak our fabulous language, don't worry, I will get back to English soon. Maar ik wil ook eerst even zeggen, Rick, wie lang kennen we elkaar al? En wat voor traject hebben we doorgemaakt? Zorgen dat voor de wisten zijn we eigenlijk heel kort bij elkaar in de buurt opgegroeid. Ja. Maar toen kenden we elkaar niet. Zeker niet toen ik opgroeide, want toen was ik nog niet geboren. Dus dat hebben we niet gekend. Maar later hebben we elkaar ontmoet en hebben onze paden eigenlijk elkaar steeds weer gekruist. En zijn we eigenlijk in touch gebleven tot op dit moment, waar ik ook heel erg gerust ben dat we dit samen kunnen vieren. Having said that, we go back to English. So today I looked a bit in the, and my memory is not that great. My computer's memories are great, right? So I look a bit in files today to come up with a bit of a history. Then I came to 2016, right? You were at Zuid University of Applied Sciences, took our bioinformatics module. And this is a module on which we teach only a few days. And there's, lots of students you don't know, and you commonly don't remember them. But some of you do remember. And I already noticed back then that Rick was very enthusiastic playing with sequencing data, with the MinION sequencing, and asked all kinds of questions for more clarification and this was really, really cool. But then, yeah, a bit later, you also chose to do your bachelor internship with us after some previous experience in other internships that weren't great, let's phrase it like this, right? And you thought it was maybe a bit of a waste of time. But I actually think it was also very valuable. You got to an insight what you wanted to do, and definitely which people you wanted to interact with, and apparently I was one of these. So this is good news. And you came to us to for an internship in bioinformatics at the time, and I think you really flourished there. You found your domain and you found the things you liked. So I also got to know you at the time as a tech freak a bit, right? You always had something you were fiddling with at home, and you wanted to show me pictures or real things you brought, right? I think of whatnot that you make. I think you had your LED lights that you programmed to do, I don't know what all they did. So you came up with 3D printers that you built yourself. You brought a drone and you want me to come out to see you fly it, right? And I thought, okay, this is all cool. And honestly, this is the kind of student that brings back your own history as a student a bit. You know, at some point we get mostly bored with administration and things, right? But you also have a passion for Tiger Science as a academic staff. And you're one of the persons, I think, that also, you know, got me a bit back there. Oh yeah, this is cool. So very cool. And I think nowadays you even got into AI-generated like phone covers and everything. You add everything to your portfolio, right? So going back to the bachelor internship, at that time we sat you together in a room with interns from all kinds of disciplines. And I think this is something that is really valuable. And I abuse the opportunity now to maybe make also a statement for the university, like, you know, please seat students at the department. You know, students from various backgrounds, I think you learned a lot about other domains and the skills that the students learn at university. Yeah. I think at the time it also was clear that you're a very social guy, besides a tech freak, and always, you know, enjoying to engage in everything, right? And I'll get there a bit later. I think you even accepted to be in the little team I created with you and some of the peers, which we call the Pute. So for those who don't speak Limburgian, Pute means kids, but it has a bit of a connotation as in a bit annoying kids, right? And so the fact that you accepted to be in my supervisory group called this was good. So at the end of the bachelor internship, we also went to some conferences, right? So we want to give you the opportunity, and one I recall is MOSA conference. It's in Maastricht University. It's for our own students, so to say. But I thought, it's for master students mostly, but I just signed Rick up as an external student. But what happened? He won the award for oral presentation. As a University of Applied Sciences student who'd never done such a thing. So I think you sweat like a pig, or like an otter, as we say in Dutch, at the time. So very nervous, very different from now. But it was brilliant, right? First lecture. Then we went to some other conferences, BIOSB, Dutch Neuroscience meeting, System Biology meeting, right? You took a poster, and I clearly saw you liked engagement with all the audience. You also liked the partying and the very nice bits, right? And the nights. And fortunately for you, I also like these. So my memories of that is very, very blurred. So we can't go there. Okay. Then you came back for your master internship. For your master, you did the Master of Systems Biology, and I think this was a well-informed choice now in the right domain, right? Yep. Exactly where you wanted to be. And you did your internship again with us, with Supervisor Rachel Cavill from, at that time called, I have to read that, Data Science and Knowledge Engineering, which is now DAX, and at our Department of Psychiatry and Neuropsychology. And you completed the thesis, Prediction of Post-Traumatic Stress Disorder Risk in Pre-Deployment Military Servicemen, with a very good result. And you already hear the similarity to the topic now. So we hired you as a PhD candidate, right, on the vacancy we had on Professor Rutten's VD funding. So, you loved the machine learning, that was already clear. So our data didn't always fully allow that, right? So that's why also answering to some of the questions you said, it was a bit pragmatic that I used various conditions. Let's see. What you also developed into was a key member of your PhD room. I think when we came in for some meetings, it was always clear it was very nice there. It was a nice atmosphere. And I think on both sides, first of all, it showed in the science, because you contribute to many other people's papers and were consulted, given advice, which we also see in the thesis. But furthermore, I think it was also always there was a nice atmosphere. We can call it chaotic or, and then I think, we don't even know half of it, right? What happens there when we weren't there. Two short more things. Of course, I still wanted to address another achievement Rick made. Besides all the things he was doing, he managed in a very effective way to find his new wife. So this is also a great achievement. I think this was so cool. By the time we found out he had a new girlfriend, and then he had a trip to Pakistan, to see the family. We also were, by the way, that he also would get married there. So we were a bit like, okay, so we know. That's good to know. So the final thing before handing over to Aston for a brief bit on as a follow-up is to warn you. Because one thing you also took up strongly was participating in the roasts of the previous graduates. You were always one of the people very engaged in roasting others. And well, as we say in Dutch, wie kaatst moet de bal verwachten. So I wasn't involved in that, but what goes around comes around. So I expect that this evening you might suffer. But for that to see. So for a last line, go back to Limburgian. Ja, nogmaals proficiat, Rick. Ik denk dat er een prachtige toekomst voor dich ligt met allerlei mogelijkheden. De kids, de academieën, de industrie, eigen bedrijven, alles ligt voor dich. Daar heb je de helft al gedaan. Hè? Je kunt alle kanten op gaan en ik denk ook met de prachtige nieuwe Sean-familie. Ziet het er allemaal fantastisch uit en daarmee geef ik over aan Essel.
Thank you very much. So, mine is short. So, dear Rick, I still remember how impressed I was by your enthusiasm and effort during your internship. So much so that after you submitted your thesis, I asked you if you would be interested in joining a project where we aim to apply machine learning to study cognitive function in Parkinson's disease. At the time, I thought it would be just some brainstorming about methods, nothing too big. But then things took off. Together with Josh, who was based in the UK, we started making real progress. Not long after, Bart invited me to join your supervision team. And then, of course, COVID happened. We were all stuck at home trying to make sense of a very strange time. But in the middle of that, something great happened. You became a key part of that project. When I first invited you to one of our Wednesday 10 o'clock meeting with Josh, I had no idea how much you would shape the project or how much fun our collaboration would be. Your energy and passion were contagious. In fact, sometimes your enthusiasm was so high that I had to stop you from reinventing the wheel.
Yes.
Along the way, we all learned an important lesson. Science builds on what came before. We had to take the time to carefully study previous research and cross-check every new idea against what was already known. We realized that any novel approach must be backed by a strong reasoning and a clear understanding on how it moves the field forward. That balance between creativity and careful groundwork is what makes great research. I'm very happy that you joined the ADPD conference in Gothenburg as well, leaving us with unforgettable memories. And of course, I will politely skip over the first evening when you decided to interrupt my conversation with your friendly expression. Rick, today we celebrate not only your hard work, but also the person you have become through this journey, a dedicated team player, insightful researcher who will no doubt to continue to make an impact. Congratulations to you and your loved ones.
Thanks so much.
Dear Dr. Reijnders, I also want to congratulate you, and I have the honor also to do this on behalf of the community of Maastricht University. You did an excellent job, and I wish you a lot of success in your future. I want to give the floor to Professor Homberg for a short word.
Well, congratulations. I really enjoyed your thesis defense. I also liked your introductory talk because I'm not totally in your field and I could fully understand it. I can see that you are highly talented and I wish you a lot of luck for the future.
Thank you so much.
Dr. Daskalakis.
Congratulations, doctor, and I really enjoyed being part of your committee. I knew your work in the context of PTSD and system biology, but It was a great opportunity to see all the body of the great work you have done, and looking forward to see your next steps in science.
Absolutely. Thank you. Thank you so much.
Congratulations.
Thank you.
Okay, thank you all for participating. After closing this ceremony, I invite the audience to go to the rafter for the reception. We stay a little bit longer here to make some pictures, and we join as soon as possible. Thank you for all being here, and hereby I close this academic ceremony.
Dankjewel.